everyone, my name is Eungi and I've been working on a project called Decoding Systems Complexity using Natural Tokenization with other fellow research associate Ceren, Balkan, and Kaoutar and Elasri. We have one amazing visiting scholar, Tiana Underworker, and also a lot of amazing principal investigators, Dr. Celia Blanco and Henderson Claves, and McCollins and Dora. So to begin this presentation, I'd like to ask you all a question. Like, does any of you here know which organism has the largest genome ever founded? Well, we might think of big animals or mammals like elephant or what else? Whales or even humans, but surprisingly, the organism that has largest genome ever found is the fern-like plant. And this one has a genome which is 50 times larger than the human genome. And by this result, we can say that the long genome does not necessarily mean the organism's sophistication. But here, how does this small plant contain this long genome? And how can we analyze this long information? So this is the place where tokenization takes place. And before we talk about the tokenization, I'd like to bring it, I'd like to take a wide example like this. In this slide, this looks a bit more like not really neat and a bit distracting. But if you see this slide, we can see that there are a lot of different letters. And there might be a way to compress this information or make it a bit more briefly or shorter. There could be several ways, so feel free to send the chat in the chat what kind of ideas do you guys have. But one easy way to say or compress the information on this slide is that we can say in this slide there are A, T, and G is repeated several times. So broadly, this is the big concept of tokenization. And to be more specific, we can look at this example. I prepared a tongue twister sentence. Peter Piper picked a pack of pickled peppers. Here, we can divide the sentence into smaller pieces into words like this. Peter Piper picked, and etc. After that, we can also divide the words into smaller pieces, which are repeated. So here we have PE and PI, CKED repeated. And so when we tokenize, we split the text into subwords, which is grouped, subword, and the tokens here, the subword contained in the vocabulary, which is the group of frequent tokens. And by doing so, we can process and analyze the complex data more efficiently. But here, one key concept to note with tokenization is the definition of compression. Compression basically means to store the information in a compact form. Well, we have original data like this, and then we can compress the data with specific algorithm to make it smaller in the size way. And if we compress the data, it's easier to transfer or analyze. And also we can decompress the data to the original version with a certain way of algorithm. One of the familiar example of compression is gzip. And we're not going to talk about the technical details of it, but here we can see that we have a raw data and the specific algorithm or the compression software allocate the other informations to the raw data and then compress it to reduce the data size. So how could we apply this compression and tokenization to DNA sequence? For the sequence on the left, we cannot really say that the sequence has something repeated. It doesn't seem to have something in common in it, which means we need many tokens to explain the sequence. And this means that this sequence is less compressible. However, in the second sequence at the bottom, we can see there are some repeated chunks. So here we can say there are AAG, 2 AAG, and 2 TC in the sequence. And this means the high repetition means we can we can describe the sequence with few tokens, and it means the sequence is more compressible. And before we go into detail of our result, I'd like to explain a basic principle of biology, the central dogma. The central dogma describes the flow of genetic information from DNA to RNA to proteins. DNA is composed of 4 different nucleotides, A, T, G, and C, which are then grouped into 3 nucleotide sequences called codons. These codons, in turn, finally encode the 20 amino acids that makes up proteins. The system of DNA, codons, and amino acids is a kind of natural form of tokenization. Each level represents a form of tokenization that allows a complex information to be organized and also processed. And our DNA sequence can be roughly divided into 2 parts, like coding region and non-coding regions. This plot shows the composition of Homo sapiens genome, and surprisingly, only 2% of our genome code DNA is a coding DNA, which means only 2% of our DNA makes proteins, and other 98% has some other significant roles rather than making proteins. And there are 2 important abbreviations to remember. First one is CDS, which means the coding DNA region, and ncRNA as non-coding part. Like some DNA is transcribed to RNA, but ncRNA is ncRNA means the RNA which is transcribed but does not code proteins. So between these two, CDS and ncRNA, there are different patterns in how the information is stored. And also some organisms like bacteria, they have different amounts of ncRNA compared to others.
Hello, everyone. It's great to be here, and I want to tell you about the methodology we followed. So we obtain our CDS and cRNA data from Ensemble database as an annotated sequence files. We use organisms from 6 different kingdoms of life. These are animals, archaeas, bacteria, protists, fungi, and plants. As you can see from the pie charts, these organisms have very different kind of ratios of coding regions and transcribed non-coding regions. And as an example of prokaryotic organisms, E. coli almost entirely composed of consists of protein coding regions and has very minimal transcribed non-coding regions. But the opposite is true for Homo sapiens. Yeah, and we use word piece and sentence piece algorithms for tokenization. But beforehand, we use pre-tokenization to better handle the large genomic data. And after that, we use compression algorithms. So we use chimerization as a pre-tokenization system. Chimerization is based on a defined number of K as a reading frame. And here we can see an example, which K is defined as 6, and starting from the first 6 nucleotides, this chimer shifts 1 nucleotide at a time and reads the whole long DNA sequences. And as a result, it splits DNA into manageable parts. So we choose sentence piece tokenizer or word piece tokenizer, but why is that? Sentence piece tokenizer does not search for pre-tokens, and it treats the input data as a Unicode characters, and it eliminates the need for a language structure that relies on separate words. And it is also fast and efficient, which makes it ideal for large genomes. It also minimizes the information loss because it does not need pre-tokenization. And it also generates multiple possible tokens at once and allow us to identify and handle the rare variations in genome, such as mutations. So we ended up using sentence piece and we leave chimerization because we don't need pre-tokenization anymore, and we directly compress our data with compression algorithms. So tokens aims to store more information in smaller sequence. So we can confidently say compressibility is an important part of the tokenization. And we use two different kinds of compression in this project. First, we computed compressibility on tokenized sequences, and then we compute compressibility on original sequences with the compression algorithms. We calculated compression factor for tokenized sequences, and it is calculated by adding the length of the encoded tokens by the length of the vocab and dividing by the length of the original genome and multiplying by 100. And as an example, we have 12 nucleotide sequence here. And when we tokenize the sequence, it encodes like this. And here our length of the encoded sequence is 5, and our dictionary is made from 3 tokens. When we add them together and divide by the original length of the given sequence, multiplying by 100, we can say the encoded sequence covers exactly 60% of the original size of the given sequence. And another compression method we applied is based on compression algorithms such as LZMA, gzip, and Gecko3. We applied all these algorithms to the unprocessed original sequence files. And these methods build a dictionary of repeated patterns found in the data, especially Gecko3 method developed for genomic data, and it detects deeper patterns unique to the DNA sequence, such as repetitive elements, inverted repeats, and etc. And we use compression ratio to calculate compression. It is calculated by dividing the compressed size by original size and multiplying by 100. So what are we looking for in our projects? First, can we detect tokenization patterns in genetic information? And the second question is, are compression trends conserved across different domains of life? And these are the questions we try to answer in the results.
Hello, everyone. So in the upcoming slides, I'm going to share with you the plots that we have worked on. The first plot is going to compare between tokens in coding regions and no coding RNA. So in the left, there is a graph that shows the token length, like the frequency of tokens vocabulary versus token length in coded sequences. And one of the things that we can notice is that the token length here is more unified for coding sequences compared to no coding RNA in the right. You can see they're much more different lengths and they vary differently, but this could tell us that in coding sequence, we have a conserved or like a pretty unified token length. And for the next plot, we have generated a diagram that also going to compare between coding sequence and non-coding RNA tokens. Okay, so for this diagram, one of the main difference that we can see is that no-coding RNA tokens have less common tokens to coding sequences, which is a very interesting finding that we were able to look at. And just to share with you how these plots were made, so this data comes from 10 different organisms that come across all domains. And this was generated using the vocabulary of each organism by itself. So something that could be interesting to look at is if we can try this on much more organisms and if this pattern will be conserved or would it be changing and which factor would affect its changes. For the next plot that we're going to share, it's just going to show if tokens do match between different organisms. Okay, so for this, two different plots, we comparing between tokens and fungus versus bison. So the plot in the left shows that the tokens do overlap for those who are the most frequent. So we have about 0.26, which is 26% common tokens between fungus and bison. Similarly, we can see that in the second plot, that frequency of tokens, if they're like more frequent, you can find similar tokens in other different organisms, which is pretty interesting to look at, because if we think about different organisms, we might see that like genome sequences, especially like coding sequences, might be different because organisms do tend to have different functions. But here we can see that actually the long tokens that are very frequent can be found more commonly. And this is going to be interesting if we can find the same results for other organisms. And one of the last things that we try to look at is the effect of coding material and its relationship to complexity. So we try to see if actually having much more coding material or like longer coding sequences means that it's going to be more complex or not. So here in this plot, we have 4 different domains. We have vertebrates, fungi, bacteria, and plants. We can see like in yellow, we have bacteria and they have the shortest total coding sequence length, and they have a higher compression ratio, which means that they are not very well compressed. And one of the explanations to this is that maybe those bacteria, they have very complex coding sequences. In contrast, we have plants and vertebrates. They have much longer coding sequences. That's why they are in the right. And they have lower compression ratio, which means that they are more likely to be less complex because they are easily compressed. And this bring us back to the first question that Eungi said, like asking in the beginning of this presentation, which is the longest genome? And surprisingly, it was found in a very small plant, maybe because this plant has a less complex genome. That's why it was easily compressed. And we're talking specifically about coding sequences. And it will be interesting to look at this in other different organisms in different domains and even compare between coding sequences and encoding RNAs. But let me mention, first of all, some of the challenges that we have encountered in this research that might affect the results that we have found. So first of all, we have data. It is difficult to find the right data to analyze. There are multiple websites like NCBI and Ensemble that we can find genome data in it, but Ensemble was the one that we opted for because of its simplicity and how easily it was, we were able to download data from it. And also there is the computation challenge. It's not easy to analyze large genome data. It might take a lot of time to be able to find outputs or to generate plots. So those were some of the challenges that we have encountered. But we do certainly have future directions and future things that we want to look at more deeply in this research. Okay, 1 second for the future directions. Okay, so for the future directions, we want to look up tokenization patterns across more domains of life, so we could see if the results we have are going to be the same or if they're going to change and why are they changing in case. And also we want to look at compression and tokenization in coding regions versus non-coding regions and see the more differences if they occur. And also look at tokens network analysis and see if there is a certain connection between different tokens. And yeah, that's it. Thank you so much for attending this presentation, and feel free to ask questions if you have any.
Yay, that was awesome. That was so many cool comments in the chat, but if anyone has any questions right now to ask, you can also raise your hand or ask in the chat. We can send those along as well. But I really love where the talk came to. That was a great explanation of tokenization and the work that you've done this summer. Sanjoy, you're first up.
My mind is still blown. This is just incredible. Do you think compression has changed over geological time? So if I'm thinking about some of the more primitive microbes like the methanogens or the acetogens, how does their ability to compress their genome compare to more recent metabolisms like photosynthesis or even like I guess trying to remember wrapping my head. Does evolution affect compression ability?
If I can take this question, this is just an assumption, but I think you will, because we noticed that like compression is related to complexity. And if evolution is going to change and modify like an organism's genome because that organism needs to adapt to certain changes in its environment. And if it gets more complex, it's going to be directly affecting compression. So I think there is a very big correlation between those 2 points. And I feel like it could play a big factor. We have not really thought about like comparing between certain organisms that have existed in the past and how they were able to, how to say evolve, like how they changed their evolution. But that could be an interesting point to look at. I think it would make sense that compression would change based on how the evolution happens and if the genome gets more complex, because something cool happened to that organism, a new metabolism have appeared, a new type of adaptation to the environment changed a certain parts of the gene. So yeah, that's more likely to happen based on my assumption.